Hello students, welcome to EPG Pathshala. I am Dr. Renu Tyagi from Department of Anthropology, University of Delhi. Today we will be discussing about the module Genetic Diversity among Indian Population under the paper Human Origin and Evolution. The learning objectives of this module are to understand the genetic diversity, to know about the potential factors of population diversity, to have the knowledge of composition of the Indian population, to comprehend the genetic diversity among Indian population. Now let's have an introduction about the topic. In the last few decades, anthropological and genetic studies have witnessed explosion of the human genetic data. These data have greatly enhanced the human understanding of the pattern of genetic diversity and variability among individuals and population groups. The genetic diversity refers to the heritable variation within and between the populations of the organism that serve as a way for populations to adapt to new and the altered environments. According to Brown in 1983, genetic diversity is the amount of the genetic variability among individuals of a variety or population of a species. It can also be defined as the clay of the evolution which provides the base material for adaptation and speciation. High level of genetic variability is regarded as healthy, conferring the ability to respond to the threat such as disease, parasites and predators and environmental change, whereas the low level of the variability is considered as limiting a species' ability to respond to these threats in both the long and the short term. The extent and the distribution of the genetic diversity need to be understood. Genetic diversity results from the many genetic differences between individual and populations and may be manifested in differences in the DNA sequence. In biochemical characteristics, for example in the protein structure or in the isoenzyme properties, in physiological properties, for example the abiotic stress resistance or the growth rate, or in the terms of the morphological characters, the four components of the genetic diversity can be carefully distinguished. That is the number of the different forms called as alleles ultimately found in the different population, their distribution and the effect they have on the performance and the overall distinctions between the different population. The variation that underpins the genetic diversity arise from the mutation and recombination, selection, genetic drift and gene flow which act on the allele present in different populations to cause the variations. With the passage of time, a population divides into many subpopulations because of the various cultural and demographic methods. The consequent demographic and environmental changes primarily reduce the size of the population, that is the formation of the bottlenecks, which lead to the increase in the genetic diversity among the subpopulation under the impact of genetic drift when such subpopulation remain isolated from one another, that is the individuals of one subpopulation do not exchange mates with the other subpopulation, the genetic diversification is hastened. The fusion of the population also occurs but in much less common than the fission. The primary forces that favor the genetic diversification includes the cultural and the linguistic differences and geographical distances. Thus, one expects genes, culture and language to evolve in the tandems, especially for such genes that are not subjected to differential pressure of natural selection. Due to his expected phenomena, the anthropologists are always fascinated by the correlation of genetic diversities with the cultural, linguistic and geographical histories. In general, the longer two populations are separated, the larger is the genetic differences between them. This figure shows the evolutionary paradigm of the genetic studies of contemporary human population according to the Majumdar. The classification of the Indian population, that is the ethno-cultural and geographical classification, need to be understood. The Indian subcontinent is populated by more than 1 billion people who are divided into a large number of the endogamous population groups consisting of the different caste, languages, religion and tribes. The caste system in India was formed primarily on the philosophy of the division of the labor with cooperation rather than competition. This consequently has led to a hierarchical division. The Brahmanas, who have the traditional occupation of preaching, belong to the top level, and so-called the scheduled caste, who are supposed to perform the menial tasks such as scavenging, occupy the bottom level. The religion of all the caste group is Hinduism. The tribal groups do not have a uniform religion and many are ancestor worshipper. The major language spoken by the caste group of the northern half of the India belong to the Indo-European family, while those spoken by the caste group of the southern half belong to the Dravidian family. The tribal population of India are supposedly the first settler and their number is about 645. These tribes are scattered all over the India and vary greatly in their numeric strength. Linguistically, they belong to and of the four broad linguistic families, that is Austroasiatic, Khasi group, Santhali, Ho, Mundari, Gonds, etc. The Dravidian groups, for example Korva and Yerkula, etc. And the third one is the Indo-European group, for which Bhils is an example, and culturally they can broadly be classified as the hunter-gatherers, hill cultivators or agriculturists, artisans and industrial laborers. In some urban cases, apart from the caste and the tribes, there are several other religious groups, that is Hindus, Muslims, Christians, Parsis, Iranis, Sikhs, Buddhists, etc. This table displays the morphological types, features and geographical distribution of people of the India, which include Negrito, Australoid, Mongoloid and Caucasoids. The Negritos are the short stature, frizzly hair, fine hair texture, brachycephalic head, dark complexion, short and protruding face with broad nose, thick and everted lips. Their geographical distribution is from the Andaman Island to the Nilgiris of the Tamil Nadu. They may have had a wide distribution at one time. The next group is the Australoid, who are the short stature, taller than the Negrito, and they have wavy to curly hair. They are hyperdolichocephalic to dolichocephalic, with their head, dark complexion, stout brow ridges with sunken nasal root, spread chiefly in central and the southern region. The Mongoloid groups are the short stature with broad shoulders, scanty facial and body hair, who are brachycephalic. They have the flat face, prominent cheek, bones, flat nose. They have epicanthic fold spread over the sub-Himalayan and northeastern regions. The next category is of Caucasoids. They are generally tall stature, dolichocephalic to brachycephalic. They have the light complexion. They have straight to wavy hair, sometimes light eyes, arched forehead. They have long face with well-developed chin, narrow and prominent nose, and they are found in most of the region. Throughout the history, many population groups migrated successively to India from north, west and the northeast. It is believed that the ancestor of some existing tribes in South India entered the country from the northwest in prehistoric times. Some of the migrant populations maintained their genetic identity while others were dissolved by the interbreeding. With the general population, cases are not rare that population living side by side for hundreds or even thousands of years retain their genetic differences by practicing endogamy. Biological anthropologists believe that the breakup of the social isolates leads to an increased incidence of the heterozygotes of dominant types in the result population. The endogamy of the religious groups leads to the differentiation of the people. All of these cultural, linguistic, religious, morphological and migrational factors have affected the gene flow and genetic diversity within and between the Indian population. Now let's understand the genetic diversity among the Indian population. For many years, anthropologists studied the problem of differentiation in Indian population by archaeological or linguistic evidences as well as by the fossil records and morphological characters. The conclusion reached by all these evidences are not in complete agreement because of fragmentary nature of the fossil and change in the morphological characters due to environmental factor. Therefore, genetic approach is used to study the differentiation and diversification of the human population, which is stable and non-susceptible to environmental factors. According to the Kapoor in 2000, genetic studies of the population from the Indian subcontinent are of great interest because of the India's large population size, complex demographic history and unique social structure. One measure of the genetic diversity is what is termed as the average heterozygosity H. The average is taken over a large number of the genetic loci, that is L. The heterozygosity at ith locus, where I can be 1 to nth, is defined as where Pi denotes the frequency of the ith allele at this locus with N1 allele. H1 is a measure of the proportion of the individual in the population who are heterozygous at this locus. There is extensive genetic diversity not only at the all India level but also within the smaller geographical regions of India. This figure demonstrates the average heterozygosities based on the data of the variable number of loci in some defined ethnic groups from 4 geographical regions of the India. The next figure displays the dendrogram depicting the genetic relationship based on the data of the 10 loci among population from different geographical regions of the India. The further figure shows the dendrogram depicting the genetic relationship among population of the eastern India. And the next figure, according to Majumdar, shows the dendrogram showing genetic relationship among the population of the southern India. Some of the important studies conducted on the genetic diversity among Indian population need to be studied in this context. Genetic diversity among South Indian tribal population, as studied by the Watkins et al., they investigated the genetic diversity among the 9 tribal population groups of the Andhra Pradesh, Karnataka, Kerala, Madhya Pradesh, Orissa, Tamil Nadu. An 8 caste group of the Andhra Pradesh, Southern India tribal population demonstrated low within population heterozygosity within the range of 0.54 to 0.69. While tribal populations sampled further north and east have higher heterozygosity within the range of 0.69 to 0.74. The genetic distance which estimate showed that the tribal Indians are more closely related to the caste Indians than the other major groups. Between the tribe differentiation was found to be high. Telugu speaking populations were less differentiated than the non-Telugu speaking population, where the fixation index shows the value of 0.029 versus 0.079. The South Indian tribes showed significant population structure and individuals clustered statistically into groups that corresponded with their tribal affiliations. These results were consistent with the high level of the genetic drift and isolation in Indian tribal population, particularly those of the South India. This figure demonstrates an unrooted neighbor joining network of the 35 population based on pairwise genetic distance DSW estimates using 45 STRs. An inset indicates the relationship for the major population groups. Now let's see the genetic differentiation among ethnic groups of the Punjab. The state of Punjab in the northwest part of the India has acted as the main passage for all the major human invasions into the Indian subcontinent. It has resulted in the mixing of the foreign gene pool into the local population, which led to an extensive range of the genetic diversity. Singh et al. In 2016 studied the extent of the genetic differentiation at 10 autosomal DNA locus such as the ALU elements and SNPs among 5 Indo-European speaking ethnic groups of the Punjab, namely Baniyas, Brahmins, Jat Sikhs, Khatris and scheduled caste. The phylogenetic analysis revealed that Baniyas and Khatris are genetically closest to each other. The Jat Sikhs are genetically close to the Brahmins and are distant from the Baniyas. The Jat Sikhs, Baniyas, Brahmins and the Khatris are genetically very distinct or distant from the scheduled caste. Overall, uniform allele frequency distribution patterns, high average heterozygosity values and a small degree of the genetic differentiation in this study suggested a genetic proximity among the selected population. A low level of genetic differentiation was observed in the studied population groups, indicating the genetic drift, which might have been small or negligible in shaping the genetic structure of the northwest Indian population. This figure shows the neighbor joining tree based on the DA or the distance depicting the genetic relationship among 5 studied population based on 10 autosomal markers as studied by Singh et al. In 2016. The genetic diversity among the central Indian tribal population as studied by Das et al. Reported that the nature and the extent of the genetic differentiation among 16 tribes of the Orissa, Madhya Pradesh and Maharashtra were found. 11 tribes spoke the Dravidian language, 3 spoke Indo-Aryan and 2 spoke the language of Austro-Asiatic. The population structure of these tribal population was analyzed at the inter and the intrastate level and linguistic level. Using data for the 13 genetic system, 38 allele or haplotypic, 9 of the 13 loci showed significant heterogeneity in the 16 tribes, and the pattern of the heterogeneity was also discernible in the different states and in the Dravidian speaking tribes. The extent of the genetic differentiation or gene diversity was found to be the highest from central India. This figure shows the dendrogram depicting the relationship among 16 tribal population of Orissa, Madhya Pradesh and Maharashtra as studied by Das et al. The genetic differentiation among the 12 ethnic groups of India was studied by the Sen Gupta et al. In 2004. They examined the pattern of DNA sequence variation in and around the gene coding for CAM1 and TNF in 12 diverse ethnic groups of India, namely Bhotiyas, Mizo, Meiteis, Santals, West Bengal Brahmins, Kadars, Iyer,s Mauryas, Saryapuri Brahmins, Marathas, Konkan Brahmins and Jarvas. The extent of the genetic variations attributable to the between group differences was quite low, and that among the population within the group was only slightly higher. At both loci, most of the genetic variation was attributable to the differences between the individual within population. This figure shows the bidimensional plot of the first two principal components extracted from the haplotype frequencies of the ICAM1 and TNF loci, depicting the relationship among the 12 ethnic groups as studied by Sen-Gupta et al. Now let's have a look at the genetic diversity among population group of the Himalayas as studied by Kapoor and Tiwari in 1986. They investigated the genetic diversity among the 16 population groups of Himalayas on the basis of 4 genetic loci. It was observed that the genetic heterogeneity within the Himalayan groups is large as compared to between groups. The high genetic distance between various castes and the Mongoloid group of the Himalayan point to the independent process of genetic differentiation in this population group. This figure 10 shows the dendrogram depicting the genetic relationship between 16 population group of the Himalayas as studied by Kapoor and Tiwari in 1986. The studies of the maternal lineage by the mitochondrial sequencing have shown that two major mitochondrial lineages that emerged from the Africa haplogroups M and N dating to approximately 60,000 years ago are both very diverse among Indian population. Additional studies of the mitochondrial haplogroups shows that an early migration may have populated the Indian subcontinent, leaving the relic population in present-day India represented by some Austroasiatic and Dravidian-speaking tribal population. These results highlights that the initial peopling of the Indian subcontinent likely occurred as early as in the history of anatomically modern humans. Concordant with the mitochondrial DNA, that is mDNA, date paternal lineages within India also show high diversity based on the short tandem repeat STR markers on the Y chromosome and support an early and continuous presence of the population on the subcontinent. Studies on the autosomal SNPs and STRs also demonstrate a high degree of the genetic differentiation among Indian ethnic and linguistic groups. A recent study done by the Rick et al. In 2009 provided strong evidences for two ancient populations, genetically divergent, that are ancestral to most Indian today. One, the ancestral North Indians, ANI, is genetically close to the Middle Easterners, Central Asians and Europeans, while the other, the ancestral South Indian, ASIs and East Asian, as they are from each other. The fact that Dravidian-speaking southern India remained relatively sheltered from the Indo-Aryan influence is probably the major reason why South Indian population are genetically distinct from the North Indian population. The Indo-Aryan speakers who entered through northwestern and western corridors diffused in all directions, receiving a check only in south where Dravidian language were firmly established. In other words, there was relatively little admixture of the diverse genetic elements with indigenous genetic stock in southern India as compared to the northern India. Now let's summarize what we have learned so far in this module. The contemporary Indian society is quite complex with multiple groups having the widely different socio-cultural and demographic history. Because of its enormous socio-cultural, linguistic and geographic diversity, the population structure of the India draws attention of many anthropologists and human geneticists. Indian demographics have been influenced by many potential waves of the migration. Since genes move with the people, the entry of the diverse genetic elements into India must be a major reason for the extraordinary genetic diversity of the Indian population. Studies on the genetic diversity among Indian population shows Indian population harbors more genetic diversity than population of comparable geographic regions. Indians genetically are between Caucasoids and Mongoloids. Southern and the North Indians are genetically quite distinct. The geographical and the unique socio-cultural differences among Indian population play a significant role in genetic diversification. Studies also show that geographical differences play a stronger role in the genetic diversification of population than socio-cultural differences. Therefore, even when two culturally distinct populations are classified by an anthropologist as two distinct endogamous groups because of a low rate of admixture between them, say less than 0.01% of all marriages per generation in terms of the genetic homogenization, this rate may be very high. Geographical distance of the habitat, irrespective of the cultural affinity, may indeed be the limiting factor for the flow of genes between population in India, which is perhaps why geographical differences are found to be the stronger determinant of genetic diversification. Recent advances in the field of the genetics permits the choice of highly heterozygous markers. Therefore, potentially much more information markers can now be screened in human population genetic studies. Such markers need to be judiciously chosen, and more importantly, such studies need to be coordinated in the future so that a minimum number of the core loci are screened in every study to ensure a minimum degree of the data comparability. A better understanding of the Indian genetic diversity and population history can provide new insight into the early migration patterns that may have influenced the evolution of the modern humans. It is hoped that studies on the diversity among the Indian population will continue so that a more information battery of the genetic markers can be used. In order to reconstruct the human evolution in India, future studies should be conducted in a coordinated manner. Additionally, a database should also be constructed. Thank you.